Good morning, everyone, and welcome to our Bellucci Symposium 2024. And earlier this year, multiple groups published their positive results of the autoforeign gene therapy clinical trials, making everyone in the inner ear fields excited. However, mutations in autoforeign gene affects around 200,000 people, just a few percent of all the cases of congenital hearing loss. So can we develop treatments for the rest 90% congenital hearing loss? Surely the answer is yes, but we first need to know more about the hereditary hearing loss. About 50% of hereditary hearing loss, the causative genetic mutations have been identified. Then what about what are the mutations responsible for the rest 50% of cases? And what genes are these genetic mutations affected? Why mutations of these genes cause hearing loss? And what are their pathogenesis? If just deliver a exogenous wild type gene is sufficient to correct the deficiencies, how can we deliver it in safe and effective ways? With these questions in mind, we focus on the genetics of syndromic and non-syndromic hearing loss for this year's Bellucci Symposium. So we have speakers from US and all over the world lined up for today's symposium, and I'm sure their talk will inspire new ideas, new thoughts today. And as the last, I would like to thank all our speakers. Thank you for your willingness to share your work, and thank you for taking time to travel here. And thank all our audience for joining us. Enjoy. Next, I will have my colleagues, Dr. Justin Reynard, to introduce the speakers.
Good morning, everyone. I hope you can hear me. Yeah. So the first speaker today will be Dr. Min-Xin Guan. He's a professor of genetics at the University of Toronto and Zhejiang University. Dr. Guan graduated with a bachelor in biology from Zhejiang University, and I hope I'm not butchering the name, but I'm trying my best. He received his PhD at the Australian National University and a postdoc in the California Institute of Technology. Dr. Guan has made significant contribution to the study of mitochondrial disease with the identification of novel mutation in the mitochondrial 12S rRNA gene associated with hearing loss. And so currently, Dr. Guan's lab is focusing on investigating the mechanism underlying the aberrant mitochondrial tRNA metabolism. Please.
There's no point. No point. I'm drawing the but I think it's I'm very grateful here, back to States. I've been for many years. I moved to China about 10 years ago. So I'm appreciate the to invite give a talk or some work in my study. Yes, I'm working for mitochondrial, so it's a little bit different. So I go. You know, everybody knows majority hearing loss is selenium hearing loss. Conductive is very about 10%. Majority selenium hearing loss is about over 90%. So etiology. Okay, so sorry. Etiology of hearing loss is majorly genetics and the environmental factors. So also we are talking about the environmental factors, very much is aminoglycoside toxicity, and also is interaction between genetics and environmental factors. So genetic hearing loss very much affect any mode of inheritance. You know, is autosomal dominant and recessive X-linked and the mitochondrial, and any chromosome. No, any chromosomes, the gene, and also in the mitochondrial. The function of the gene mainly for cytoskeleton like myosin, actin, and iron homeostasis. And I'm working today for focus on energy production, which is the mitochondrial involved. So what is the maternal inheritance? You know, people, you know, it's all clinical phenotype all transmitted from mothers. So affected mother have phenotype, affected father as no. So the question is why? Is mitochondria has own DNA. So sperm is like 100 copies of mitochondrial DNA, but the egg is oncogenesis about 800,000 copy mitochondrial molecule. During this fertilization, you know, all from sperm's DNA digest everything come mitochondrial DNA all from mother. So this is the mitochondrial all from mother. So is a good mother carry mitochondrial or bad or carry those kids. So mitochondrial DNA has own genome. I just briefly talk about mitochondrial DNA. And what the mitochondria is energy production usually, and also involve the lot of other functions like apoptosis, you know, very metabolic harbors and signal transductions. And mitochondrial genome encoding double-strand DNA, including like 237 genes, about 22 DNA, 2 ribosomal RNA, 13 polypeptide. And the mitochondrial protein, majority mitochondrial protein, 150 proteins actually included by nuclear genomes. So so mitochondrial genome is different than nuclear genomes. They are very much no intron. So only has encoded region is the loop region. So cluster, there's 2 ribosomal RNA. So and since 1988, Dr. Wallace defined the mitochondrial mutation associated with hearing is with disease. A number of mitochondrial mutation has been identified. So I'm talking about today is involved with some hearing loss, is where the larger deletion involved the precursors, and some mutation involved the syndromics. So no syndromics basically blocks the ribosomal RNA and a lot of tRNA mutations involved. So key issue of mitochondrial disorders actually is I told you before, is mutation either from mitochondrial genome or nuclear genomes that cause mitochondrial function, and then it goes to effect with ATP and the fact targeting organ targeting and because phenotype. So at the moment is we call mitochondrial deafness is no syndromic deafness. Mostly is mitochondrial mutation involved the heteroplasmy. That's the mixture with mitochondrial wild type of mitochondrial mutant molecule in larger deletions. So the major tRNA like leucine UR mutation, which involve the also is counting about 1%, 1.5% type 2 diabetes. And today I'm going to follow the no syndromic deafness, which is only critical phenotype, very much is a homoplastic mutation. That mutation is 100% mutated molecule. And today probably like 2S RNA and Tian is, and also aminoglycoside of the toxicity mitochondria in the mitochondrial ribosomal RNA mutations. So I'm just focused back to some sort of the toxicity issue. About 2 to 5% the subject exposure mitochondrial disease drugs leading to the deafness. In China, about a couple of millions people. And this is list of the aminoglycoside, very common use like usually like a streptomycin, gentamicin, and the very common here is use poplamycin is a treatment in CF patient. Let's go back, this first pedigree was found in China. It's go to, over 500 members of the pedigree. It creation aminoglycoside toxicity. This guy, Dr. Bo, this I think biggest pedigree worldwide. And until this Dr. Hu, now he's at New York University, New York College of Medical College. He did a study in China was like early 90s, about 20% of our toxicity. First person found the easily maternal hiddenness. Until Min-Xin Fisher, I think this is the first definitely gene defined by Min-Xin Fisher. And that time he was the fellow in Harvard Medical School, and he was a hematologist. He back to Tel Aviv, and a very big pedigree in Tel Aviv. Is a typical maternal inheritance, and then he doing mitochondrial sequencing found is a liposomal pure protein, and they go back to previous slide talk of the 2 Chinese pedigree all found the 155 mutation. And this is the first deficient gene has been defined. Until our lab I started work, for the function of the 155 mutation, and we collaborated with the Chinese group. We found another mutation called the 1494 mutation. That's a bigger pedigree. So what is it doing? This is the aminoglycoside is interact, act with small mitochondrial bacterial ribosome. This is A site. So compete with amino acid tRNA, the binding to this G. That's cause miscoding. So translation something goes wrong. And human mitochondria is a bit different. Here is the CG, A change the G, or C change U. This structure more better alike. And this individual carries this mutation, just like a Chinese population, so 1 dose is cause death. Question is why? And the only this drug, in fact, is the kidney and cochlea. But kidney can regenerate, cochlea is no. And the mutation is created is the binding site aminoglycoside, and we did it like a time extension and the chemical probe right here. Just this is the white type. This is gentamicin. This is dosage is 0.1, 10, and 100, 500, 100. So it's really create the binding site. No binding anymore. But 1594 mutation, they have dosages. So that's why gentamicin is very very toxic. Gentamicin they have very much no dosage issue. Like poplamycin have dosage issue. So what is clinical application? Clinical application we can, I think we did it in China with this like 2004. We have the molecular diagnosis, and then we give instruction on how to use the drug. I get to go to the next slide. So now this individual, if you don't have both exposure aminoglycoside, they're perfectly fine. So what you do, we have here like this pedigree, usually be sporadic in China. So only one, we carry the 1555 mutation, 1494 mutation, just one effect. All the effect is because you use aminoglycoside. So we do a molecular diagnosis, we give a little the CAR, this is a list of CAR. They said all maternal members, family carries a mutation. No, therefore don't use the following aminoglycoside, gentamicin. So we deliver the CAR, we do a molecular to everybody, like everybody. So you carry, you say you cannot use it. That will be very high impact in China because like this hot pedigree, right? So this is doing molecular diagnosis is a CAR. So how many people affect and impact in Chinese population? Because these two mutations like account frequency, incidence about the aminoglycan toxicity about counted 25%. We did like a large population, like about 100,000 people, definitely people. We do screening about like about 20%, and about the 5% is something that they will carry mutation, the definite patient in China. So it's a very high impact. This we can cross. We estimate about 110 million people carry the mutation. So this mutation, doing slow molecular diagnosis, can predict this with high risk and improve safety and increase the significant increase. So it's a high impact for, I think, for prevention perspective. And if we go back, look at the pedigree, because we have like 50,000 pedigrees aminoglycoside in China. And if you look at some pedigree, you find the traumatic differences. Like this is Alpha-East family, they never use aminoglycoside, and those kids deafness. And this is Chinese pedigree, only like one effect. So this suggests have suggest they have the nuclear gene involved. And we do the larger population study, intensive biochemical study, we found out that with the mutation, like Alpha-East family with affected individuals have worse biochemical as well, no affected individuals, even no asymptomatic individuals. So that's like more by this suggests they have nuclear gene involvement. So what you do, we suggest, and we do huge biochemical study. We found the mitochondrial, this mitochondrial mutation is not sufficient to cause a problem because they have mutation carried. If we don't use the drug, they perfectly fine. Also, if we don't use the typical, since the environmental factors, that's an aminoglycoside or nuclear modified involved, that's called the problem. So what do we do? That time we start look at the nuclear modifier is two things. One thing is enhancing, is worse biochemical factor caused by mitochondrial mutation, or is suppressed, makes things doesn't work. So that time we are started with do the work when they moved to Cincinnati. This was early 1999 and Cincinnati about 2002. Because I'm training was East genetics. That time they don't have like a current have whole exome sequencing, very difficult to sequence the gene. So go back to the East, we found out that the corresponding mutation in East, we have 3 genes that have been knocked out. If we knock it out itself, this is a graceful medium. If we mitochondrial dysfunction, they cannot grow, graceful grow. And there's a glucose medium. Look at this gene we knock out, they still can grow. But the double mutation with the mitochondrial mutation and the nuclease mutation, it cannot grow. So this is the mutation, this gene called the TRMU, and this is involved with the tRNA modification. We knock down, losing tRNA, losing lysine, they don't have modified anymore. And then go to kinase approach. We're screening last time about 300 pedigree on the Caucasian, African, American, and the Chinese. We only find Caucasians. Find the mutation right here. It's called TRMU. This gene is involved with the modification of the U34 condition. TRNA losing, lysine, mutation. And then we're doing, this molecular stimulation. They clearly show have some changes in structure, and also look at the change of stability. And we're doing mitochondrial, but didn't change the mitochondrial localization. This mutation, A to G, the packaging sequence. And then we do biochemical studies. As a double mutation has changed significantly. This is a modified tRNA, and this is no modified tRNA. You can look at the modification has changed, and this affects the steady state of tRNAs and also translation. Where double mutation has a lot of worsen translation factor. So that is the also we did the knockout in the zebrafish. it clearly show has effect phenotypes in the fish. This is really less in the hair cell, even in zebrafish. Another suggests that mitochondrial mutation, the nuclear gene has interactive mutation. So what do you think to do with? I'm not the first slide here. So we also found the mutation in pedigree in China because we through like hundreds, thousands of pedigrees. We can look at what's going on. So some individuals even use the drug, multiple dose of genomycin, still fine. And then we're doing whole genome sequencing, we find the modified. So we can have some modified suppression, make the deficit not happen or make a deficit happen. And next slide, I feel spend some time to talk about we have recurrent work of mitochondrial tRNAs. We sequence about 100,000 mitochondrial gene in the tRNA. It's a tRNA, whole mitochondrial genomes. And this is the mitochondrial, this is a typical tRNA structure. And then we found out the tRNA, about your tRNA is very important for the translation. And this is tRNA post-translation processing involved processing involved tRNA modification and also the amino acylation. So what we do is we find out all tRNAs about those all tRNA variants, definite patient. We typically show the material adherence and do it by chemistry. Some tRNA is nuclear modification, and some is processing, structurability, amino acylation. So a bunch of them. We sequence everything and do it by chemistry. Like one I show you, this is like tRNA 37 position, no modification, A to G, increase the increase the methylation, tRNA methylation. This is the one of them. And this is also 30 position, this position, the tRNA methylation, the change, the increase U34, the modifications, the output by chemistry. And this is like the translation aspect that we can use. There's two techniques. One technique can use post-labeling with S35 methionine, and with inhibit the amidin can inhibit the cytosolic protein synthesis. So can run page gel, graded page gel, can look at the quantity quality change of different 13 polypeptide including mitochondrial. And this is WB really kind of change. And something I'm talking about the folding, tRNA folding. This is modification as 35, 55. And this is the pedigree we found with mitochondrial and type 2 diabetes. And the mutation you can hear cause loss in pseudo-unity at this 55. If you look at the biochemistry textbook, the 35 has a pseudo-unity, but this tRNA glutamine is lost in pseudo-unity, like the pseudo-urination loss in the mutation. And it affect the structure, tetrary structure, like a 55, with 18 tetrary structure, they make the structure more unstable, and also could affect the affinity, the conformation change, like here, white type mutants. That is best. Another is charging. This is tRNA histidine mutation, because the native gene have changed, the A to G changes, so histidine never change, and the aminoglycoside, amino acylation. The top one in the amino acid tRNA, free tRNA really change. And this one is also subcellular change of the amino acylation, the tRNA 745 mutation. That was the pedigree one from Cincinnati by Tom Freeman. Not Tom Freeman, Rick Freeman. Not Tom Freeman, Rick Freeman. We found the mutation right here. Another thing we have found the effective processing. The polycystone tRNA have a process 5' 3' check. And what this one is affected the processing, like the processing here, just a biology disappear. The mutation, that's what did I tech was possible. And in Chinese population, we found later found another mutation called 7516 mutation. That's the two transcripts, the right heavy string and right string. Right string is tRNA mutation actually localized. The heavy string is gender of the leucine UR, and the not right string, heavy string is the gender tRNA asparagine. And what we did is a 5' thymine processing, is with RSP, 3' processing. You can look here, the process defect less reduced. This is the product. The mutant is the product, this is precursor, and this is the product to tRNA effect. And look at the steady state level of all like 6 tRNAs has effect, but right string, heavy string only one effect. And also affect another precursors like the COX. Yeah, this is precursor, this is mature form. Yeah. So this we propose affect the asymptomatic effect for the tRNA process. This we did a processing effect. Another one affect the DNA DNA replication is the mutation right here as involved the replication origin effect. Origin right here, and that's what we 2D gel is looks some difficult. Look at the Fox is the effect, and look at all translation DNA replication machinery subunit. Comes like poly A, poly gamma, single cell binding protein and the helicase effect. Now look at the neurosynthesis tRNA. That's a look at BRD incorporate mitochondrial effect translation and replication. So another area proposed, all stuff with the mitochondrial. This I talk about this no syndromic mitochondrial mutation is not sufficient to cause a problem. And I talk about the really nuclear modifier or environmental factor working together, bring us to the threshold. So as very important for clinical presentation is the threshold. What level, how much is it get? So we propose about 50% decrease of mitochondrial ATP production. So 155 mutation, it only caught like 25%. And the nuclear modifier contributes another 30%. So all put together cause problem. So what is the major question they remove is the tissue specific. The question is why only cause death, not others. So we don't have animal model, mitochondria is difficult in a model. So what we will do, we're doing patient-derived iPS and using reprogramming definition the progenitor is a hair cell, it's for a ganglion cell. So our lab established a whole thing I spent like over 6 years and about 8 years to do it. It's so difficult to do it. And so we're doing what you would do is a pedigree. The lymphoblast cell lines, pedigree from Nathan Fisher pedigree, lymphoblast cell IPS, the progenitor, IPS and then progenitor definition here. And then when you increase CRISPR-Cas9 technique, crack the modified genes. So that is, use 155 mutation we did. So what you do, this pedigree, there are 4 genotypes. One is the wild type, one is the 155 mutation only. The one is like a mitochondrial mutation, one is modified mutations. Another we use CRISPR-Cas9 technique to crack the mutation into IPS. And this is to the progenitor, So the progenitor, this is 155 mutation only. This is the 155 mutation with mitochondrial, with nuclear modified, and this is the genetic correction. And then we really definitely affect the differentiation. If we have minor defect, but the double mutation is a significant defect. And look at the, if you look at the, this is a hair cell. This is white type. The 155 mutation is some defect, but not significant. And the double mutation, as clearly as morphology has changed so much. And when corrected the 155 mutation, it can some sort of rescue. Now that's all here. So let's see, look at the mitochondrial morphology. Mitochondrial use model check. There's a white type. There's a 155 mutation. Double mutation. Morphology change. That's a kind of recovery. So another evidence shows the nuclear modifier is doing the job. And then doing electrophysiology, because the big issue is really heterogeneity, you know. Look at like a clear cut with like a hair cell. But we have really good for definition is spiral ganglion cell is much better, very easy, much better to differentiate, more homogeneity than the heterogeneity hair cell. And also we did a lot of transcriptional analysis where they show the enhancement of gene functions. So another is we're doing some sort of tissue spread. Another issue, I think the translational is what is it doing? Mitochondria, does anybody can do effective treatment? Our mitochondrial field is some question, you know. One is calling to do like a metabolic, like a lot of mitochondrial disease, you do the like some metabolic, like keratins, you know, some the Q10 are doing, you know, and some you can do either can improve the treatment or can reduce the mutated molecule, or can stem cell. It could be impossible with gene therapy. You know, that's the whole thing, the gene therapy for mitochondrial deafness. What I summarize, even mitochondrial DNA mutation is, you know, is maternal inheritance, but the nuclear genes is about the maternal inheritance. And here's multiple copy. It makes things very difficult to multiple copy. Even though like CRISPR-Cas9 can do genomic editing for the mitochondria, but it's still not precise what you're doing. Like we don't have anything like pathogenic mutation. No, at the moment, no any pathogenic mutation has created is a cell line by CRISPR-Cas9 technique. So either three ways. One way is to get blocking the inheritance called the mitochondrial replacement. Another is the macrotalent technique is reduce, get rid of mutated molecule. In other words, for syndromic deafness, like Miller's mutation, 3-phosphate mutation. Those are very measured by Carlos Morales as a Zhejiang, you know, at Miami University. Now can do like use DDA can crack the mutation, but at the moment it still doesn't work. For the nuclear genes can, I think very specifically nuclear gene, the CRISPR-Cas9 correction could be very important. So I just could a little bit talk about the 3 parents baby, you know. So what you can do is like they call the mitochondrial donation treatment. You can from mitochondrial eggs, bad, you know, eggs, and with is a 2 way. One way is you know, with and the parents with the mitochondrial is good mitochondrial takes the nuclear and get a donation from a mother, the cytosol, and then make the parents genes and make a donor gene. Make it if one mother contribute bad mitochondrial contributes nuclear, and one mother is contributes the cytosol. So make it and from one egg and one spawn from the father. That will make it be 3 parents baby. The 3 parents baby, 2 mothers, one kids. You know, the father contributes the part of nuclear DNA, and one mother contributes another one. So that's going to make things work. I think the British as the first baby was born by about 5 years ago, 6 years ago, was by New York, the Hope Clinic was did like called Lee's syndrome. What you do did it is the Diwana, people moved to Diwana, did and the baby I heard is still fine. So and the British also has a baby, you know, and also Greek. I think that's could be one way you can do it in the field, you know, could broad inheritance, you know, don't let it happen, stop the maternal inheritance. So it's a big issue is positive efficiency, and another issue how the safety. Efficiency is very important. The mitochondria have DNA mutation, like Miller's mutation, when they mix together, mitochondria and the nuclear and white type and mutant mix together. Some mitochondrial DNA mutation has a replication advantages. So mitochondrial replication faster than the wild type. So even 1%, this can be 1% mutated molecule. So some like diabetes, Miller's mutation up to 5 generations could up to 50%, 60%. So another case is talk about residue issue. So the residue issue. So this is technique wise is if you use a carry, get a nuclear, you couldn't carry, some sort of mutated molecule. Was one major issue. Another safety issue. And how does it make it work? Because they have reprogramming, like you mitochondria go to nuclear, put it together, just have part of how reprogram. What does it happen? What do you know? We still don't know. So I think it seems that, but people have done a few of them in nuclear, but mitochondria definitely still nobody did. So another case with open question, we can do possible, there's another approach for the translation. So I'm to talk about where I'm from. People will talk about where I'm now. I'm in Hangzhou, right here. Very cool situation I've had. Like San Diego to Los Angeles. So I'm really here right now. So that's my group of people. Okay, thank you very much.
Thank you for the presentation. So we will start first by online question, I believe, and then we will take the question in the room, sorry. And there is 2 microphones for that.
Thank you so much for the great presentation. There's one question that interests me is that outer hair cells are more susceptible to damage than inner hair cells, especially in the autotoxic through aminoglycosides. And since mitochondria are more abundant in the outer hair cells, what are your thoughts on that? And a follow-up question is that in the iPSCs derived cells from the patients, when you look at the inner hair cells, did you take a look at the outer hair cells too to see how they are difference in their structure? And another question is that from the patients that you have collected clinically, did they have any differences in the DPOEs and ABRs? And if you know that, and thank you so much for answering.
Okay, so majorly they affected the patient only affect the hair cell, not affect the spark ganglion. This affect the, so that's one. Number 2 is the differentiation, how is IPS differentiate hair cell? It is very difficult. So you below the medium, you cannot add any aminoglycoside and any antibiotics. Like usually group DM medium added ampicillin, the double, it doesn't work, completely gone. Another thing is that with the mitochondrial mutation, this IPS differentiation is extremely difficult. We have to add a supplement and what use like a uridine, polyate, other things you cannot very difficult to differentiate the group. And number 3, we have the nutrient, have some special nutrient for IPS differentiate hair cell. We extract from egg, chicken's egg embryo, some nutrient carry and put it in this medium, it works. Otherwise, it just doesn't work. It's very difficult to do it. It's not easy to do it. And look at the if you look at the spark ganglion definition, it's much easier. So and so tremendous heterogeneity. Doesn't look like your hair cell is very easy. Look at the mice is become very different. So different. So some it took me ages to get to do it. Finally, if I work out, and all labs, not only for mitochondria, all lab first as a lab to doing CRISPR-Cas9 for patients, like we have the Min-Xin 15 and Min-Xin 7, 15. I want Min-Xin, a few of them just crack the CRISPR-Cas9.
I'm sorry.
Okay.
So.
Okay, thank you very much. Thank you.